Okay, welcome to the first video of the microbiology lab. Now, since this lab is online, we are going to be doing the best we can to go through all the scenarios. And yes, part of what you would be responsible for knowing are what are called the rules of the microbiology lab. And if I'm going to go through these very quickly, and if you'll just stick around after this, there is a virtual tour of a micro lab. It's not ours, but it does have the same things that we have. It's a little bigger than ours, but ours is actually nicer. So I'm going to start here first. Now, every culture and specimen should be regarded as dangerous. If you work in a micro lab, yes, you're working with live microbes. Now, they are attenuated, which means they are weakened. They're not the real thing. We work with Staphylococcus aureus, and the real thing would be a little more dangerous. Not dangerous. I'm not sure if the word dangerous is a little dramatic. Definitely have to be more careful with the real microbes. But still, you have to be careful. You do have to dress appropriately, a lab coat, no open-toed shoes. The idea here is to minimize the exposure to any kind of skin. You can wear gloves. No eating or drinking at any time. Of course not. You're working with live microbes, so you don't want to be eating something or drinking something, because then those microbes will end up in your mouth and then inside your system. And there's a few things here. You want to make sure you keep hair pulled back. Not a big worry in my life at the moment. If you have longer fingernails or cuts or things like that. So each day, disinfect the desktops before and after you leave. Absolutely. And you'll see that in the virtual tour. You have a lady that's cleaning off the top to show you how to do that. Ironically enough, yes, you want to try to keep things away from your mouth. Don't touch your face if you can avoid it. Always wash your hands before and after you leave, and that's a very important one. That's one thing you definitely learn in this course, is that you do wash your hands a lot more if you didn't already. Keep all wounds covered, because any time there's a break in skin and the easy entryway into the body. You will see this in the virtual tour. You really don't need a lot of items. You just need your notes and what you're working with, something to write with. You don't want to have textbooks and things, because anything that could potentially get contaminated would have to be autoclaved, which means put inside a pressure chamber of heat and steam, and that would ruin anything. So you want to make sure you just keep it to the minimum as far as what you're going to be using to take notes, that kind of thing. All contaminated materials, they must go into a biohazard bin. So and test tubes, yes, should never be placed, should always be in racks. You don't place them on the table, they can roll off. And that's kind of common sense things. In case of an accident, and you'll see this in the virtual tour, person shows you how to clean it up. She really did a very thorough job. I expected her to detonate some kind of Tony Stark device to incinerate the lab after the spill. But yeah, you want to make sure the people around you do not become contaminated. Notify the instructor if you do happen to spill something, and then disinfect the area, and you'll see that. So basically, you just use common sense with any precautions. And aseptic techniques, we will go through that. I'm going to have to find some videos to show you, but that's basically following techniques that prevent contamination of you and the cultures that you're working with. Okay, that is a quick tour. Now, if you will stick around, the virtual tour is next. All right, we're going to take a virtual tour. This is not the Howard College micro laboratory, but we can get close. So let's see here where the start is. Now, this video, this person has, well, the school has posted a sign here. It says, tie back all loose hair and clothing. That's actually a micro rule. No horseplay. Absolutely. There are no horses allowed in the micro lab whatsoever. In all seriousness, because you are dealing with fire, it's a shame we can't do this in person, but you're dealing with live microbes. That's not the time really to be playing around. Wear safety gear when instructed. We do that. We usually have students wear lab coats, no open-toed shoes, that kind of thing. No food or drink and gum at any time. And we actually have in the corner little cubby hole areas where you can put your books, and I'll sometimes let students keep drinks over there. But when you're dealing with Staphylococcus, Escherichia coli, those kinds of microbes, you don't want to be working with those microbes and then take a drink out of something or eat something, because it's going to end up in your mouth. So I'm going to hit play here. And it says something about, on the bottom, something about the hazards plaque. Maybe that's for chemicals. Who knows? Oh, see front door. Okay, for hazards plaque. All right. So we don't have windows in our lab. We do have these countertops though. And I looked in the back there. I thought I saw an incubator. She looks like she could probably use some coffee. But what she's saying here, and I'm going to pause this, what you would do in the micro lab when you come in is you would clean off the tabletops. Because, once again, we would be working with live microbes. And then here is a burner. Gosh, the name hasn't been that long. I've been up there. It's already escaping me here. Little heating plate. There's the sinks. We have all this stuff. Our sinks are actually located on the sides. And our tables are in the middle. But anyway, she's showing how you disinfect the tables when you come in and also when you leave. So next, what she's describing here is, oh, her name, okay. All right, next she is describing how you want to wear shoes that are not open-toed. You want to minimize the amount of skin that's available. There are gloves. We do use those as well. But you can see she's wearing a jacket and she has her arms covered. I mean, the goal is to minimize the exposure to skin. Once again, you're working with live microbes. In fact, I can see in the back here, if you look over her left shoulder, that looks like a biohazard fridge, which we have also in our lab prep room. And that's where all of the microbes that we're growing are kept inside there. That's what the red... Now, these are blank slides, and we will be going through, I'm going to go through scenarios with you, is what we would have done with these in a micro lab. But you can see there, they're 100% clear. And when you do staining, if you want to look at something, you start with a blank slide. Now, those are micro covers. We really don't use those that much. You would put those on top of something that you've placed on top of the slide. But if you're doing staining, you can't really put those cover slips on there. All right, now I have taken a lab course where the drawers were locked and you had a key to unlock your drawer. At Howard College, we do not have that. Here are microscopes. We have a microscope bin as well. Now at Howard, our individual tables have a microscope cabinet in front, and this in the back looks like an incubator. And I see she's got some pond things back here. You can do that. But see, here are some test tubes. There's the rack that holds the test tubes. When you work in a micro lab, you want to make sure you always keep the test tubes in some kind of bin or something. Otherwise, they will roll around. And we use the racks like what she has there. There's a petri dish. But you want to keep it where things don't roll off the table. Yeah, see that setting, 37 degrees Celsius, that's 98.6 degrees Fahrenheit, that's body temperature. And the incubators actually speed up the growth process. You can put cultures in there and leave them 24 to 48 hours and they will grow at a faster rate. Whereas it might take a week with them sitting out at room temperature. And there's the biohazard, the red biohazard. This means you have creepy crawlies in there. You don't want to be putting your submarine sandwich or anything in these fridges because that's where you have live culture. And she's obviously critiquing the refrigerators at this point and which one is better. Here is the eye wash station. If you were to get something in your eyes, we have one of these in the corner of our lab. You put your face across that and it will wash everything out. Luckily, that's never happened where we've had to do that before. Looks like here she is about to, and I don't know, I'm going through just like you guys are. It looks like a lab prep room. There is a much larger, and that looks like that could possibly be an autoclave. Let's see, ours is much smaller. Okay, there's another wash station. You can do the whole body wash there. Kill area to be autoclaved. Yes, we have that as well. Our autoclave is very small. Can't kill that many things at once, but an autoclave is something that uses pressure and temperature. It's 121, 15, 15, 121 degrees Celsius, which is super hot. It's like 212 degrees Fahrenheit. I'm not sure the math's 100% on that. But it basically kills anything. And she's describing how you would loosen the caps on the tubes when you put anything in an autoclave. Risk there of them exploding if you don't. You want to be able to let the pressure off. And slants and deeps. An auger slant, just like it says, auger slanted the tube. A deep is just a tube full of auger. We'll go over that later. Now she's going to describe taking off any labels, I would think. Once again, we're all going through this together. But that's usually what I would do, or a lab prep person. We would take off the labels before we put anything in the autoclave. Which, you know, in the end, does it really matter? No, because you're basically, after you nuke everything, you're going to throw it away anyway. And it looks like that's what she is doing. That's interesting. It is biohazard, but what we do is we put all the plates in a bag, and we take that bag and we nuke the entire bag in the autoclave. All right, she's showing how these hot plate works. It looks just like the one we have here. Not really sure what she's doing. Let's see. The suspense is killing me. Looks like another incubator in the back. We do have these sinks. Now, she's basically saying do not throw things in the sink. You want to dispose of them. There's a broken glass container. We have all that as well. And you would put tubes inside of the bin for the tubes, and you would put Petri plates in the bin for the Petri plates. All right, here is a Ventahood, which you can put things in there, and the air is siphoned up through some filters. We have one of these as well. It's not very big. If you're working with fungus, spores, things like that, you want to do it under a Ventahood. That way the spores they move up in through the Ventahood into the filtering. This looks like that's some kind of a gram stain station, which that's pretty cool. We'll go over that later. A gram stain has 4 steps to it, and this looks like all the reagents involved. I see a microscope cabinet in the background. And in case of a fire, please grab the fire extinguisher. And if you want to go deliver a pizza, there are the bags as well. So we have, fire blanket. She's just going over some basic how to dispose of items. We also have a jar where if we do some staining on our slides, we put the slides into the jar and either autoclave that or just directly throw them away. And it looks like she might be, here she's describing how you do not want to, well, in case you have a spill. If you have a spill, you want to make sure you tell the instructor. You want to keep your lab partner safe, inform them, and then clean up the area with the help of your instructor. This is why you keep everything in test tubes. It'd be really cool if they did an outline, like a body of the test tube on the table, you know, a little white outline showing where the scene happened and there was an accident or possibly a murder of the test tube. We actually use, that looks like a Bunsen burner. Well, actually, she's describing the process of cleaning. Went over this in the rules. And it looks like in case that was broken, it looks like she's putting that into a can. And then here she's cleaning up the mess. Because when you're working with anything, if that were a sterile tube, you wouldn't have to be quite as careful. But the fact that most of the time you're dealing with live organisms, kind of ironic question this day and age, but and they're really taking precautions to make sure everything is cleaned up properly. Next, I expect her to show up with a mask on and a flame floor and eviscerate that table that was spilled on. Let's see if that happens. Nope, just did a good job of cleaning. And looks like here she's taking off her gloves. Going to dispose in the biohazard. Yep, and now she's washing her hands. And that's very important. Anything else, if you learn nothing else from taking a microbiology course, and it is a popular subject nowadays, is to make sure you wash your hands. All right, thanks for joining me in that virtual tour. Okay, first virtual simulated description of an experiment. The effectiveness of hand washing. This is a very common experiment that is done in a micro lab. And basically what it involves is agar plates, which we will get to that later. That's called media. But agar plates, agar is hard jello. We buy it in powder form, mix it with water, boil it, autoclave it, and pour it in the plates. It's a sterile medium, and the microbes love it. They grow exponentially on agar because it's loaded with nutrients, and they grow into colonies, which they will not do on hard surfaces or most things because it does not support growth like that. So here's some background on hand washing. So during fetal development, now skin is sterile for the most of that process, but however, bacteria start to colonize skin during fetal development towards the end. And after a baby is born, more colonization of microbes for the first few months. That's why babies are more prone to getting rashes, because they don't have what is called normal flora established yet. So human skin is covered. A human's about 75 trillion cells. For every one cell, there are 5 bacteria cells. So we are loaded with microbes. These are very common terms for microbiology. Normal flora refers to microbes that are a permanent part of skin. And also, and it doesn't say it here, they're part of the body as well. So not only skin, we're talking about skin because we're talking about hand washing, but microbes are found everywhere. Internal organs, everywhere. Majority micro on skin is Staphylococcus aureus. That is Latin for golden. The aureus part, staph, refers to a grape-like cluster of cells. We'll get to that later too. Transient flora is what you would pick up during the day. This is what is washed off when you're washing your hands, showering, hygiene, things like that. So transient flora is not permanent. Normal flora is always on skin. We're going to talk about skin temporarily for the moment. Transient flora is parts that are not permanent. They are transients, and that's the part that you want to wash off as best as possible when washing hands. So a little background, a guy named Dr. Ignaz Semmelweis. Now you're going to go over him in lecture if you're taking lecture, but if you're not, let me just show you. Here he is, and he usually looked constipated. He apparently was a little bit of a surly guy, meaning, and he was a doctor back in his day. Was not a very good communicator, shouted a lot. This is him as an older person. But there was something called puerperal fever, which was a strep infection that was very common back in the day. Really contagious, transferred by direct contact, meaning person-to-person contact. And back in this time, and it says here 1846, yeah, about in that area, people would go into the hospital, they would get sick from this fever. Very common, because back then medical personnel did not wash hands between patients. So what he decided to do, he started making everybody that was under his command, nurses, medical personnel, they had to wash hands between patients, and he was able to document a drop in the incidence of disease. The only problem is he was a terrible communicator, he shouted a lot, and he actually was eventually committed to an asylum, and he died of puerperal fever, which is ironic. But he was not a very good communicator. And this looks like it's showing, so that's him older. And that over there looked like some medical beds. But he is the pioneering man, and he really invented hand washing. And he was able to show that after he died, people went and looked at his research and said, wow, he knew what he was talking about. Another couple of things. Skin cannot be sterilized. Skin has contours, folds, and there's a layer of oil that are produced by sebaceous glands, so skin cannot be sterilized. It can be scrubbed, transient flora taken off, some normal flora, but it cannot be sterilized. So here are agar plates. We're going to go over this later. The lid is here. The bottom, okay, the lid's the top. This area there is where the agar is. And these are sterile plates. Sterile meaning there's nothing living on the plates. Now, what we would do, and if you'll stay tuned next, there's a virtual demonstration of someone doing a hand washing experiment. We would divide the plate on the back down the middle. We have a non-wash side and a wash side. So basically this side, and what you would do is pair up together. One person's going to test their 3 fingers. The other person's going to help them do this. You would touch 3 fingers on this side with no washing. You would then go and I believe I still have it here. I do. See, these were the assigned procedures. Now, obviously, we're not going to be doing this, but like we had one group do betadine as far as the washing part. Others do regular soap, an antibacterial soap, hand sanitizer, regular water, blah, blah, blah. So you would touch 3 fingers on this side. You would then go wash hands and come back and touch the 3 fingers here. And then next, and I hope you picked this up in the rules video, you would put this plate in an incubator. It's set at body temp and wait 24 to 48 hours, which would probably be the next time you came to class, and you would then take it out, and then you would look at the results. And most likely you're going to have, where the fingers touched here, you're going to have little colonies, because when you put the fingers on the agar plate, you're putting bacteria cells on the agar, and like I said, agar makes bacteria grow exponentially. Those cells would turn into colonies, which are about 1 million cells. So if you did it right, probably going to have more growth here, a little less growth on the wash side. So that is our hand washing experiment. Now, if you will stick around next, there is a virtual hand washing person doing this experiment. All right, welcome to a hand washing demonstration. We would have done something very similar to this if you were in a lab. Love the guy's lab coat, but here's the first thing. Now, when you work with Petri plates in a micro lab, you do a Sharpie and you're going to work on the agar side. So the lid is down and the agar side is up. Now, our experiment, we basically do a before wash and an after wash. So we divide the plate down the middle. This particular group, you have access to this video if you want to watch the whole thing in its entirety. They divided it into, it looked like quarters, and they did a left and right finger before and after. So what he's doing now, and this is very important in a micro lab, you want to make sure you label everything so you can read out the results, and that's what that's called. When you grow something, you go get your plates out of the incubator, and you come back and you do what is called the readout of your plates. So he has now labeled everything appropriately. Now, see, he's taking, there's the lid on the left, he's taking his fingers, and he's going to do a before wash. So that's what he's doing. He's placing his fingers on agar. Agar is like Jell-O, a little harder Jell-O. And you see he closed it up quickly to prevent contamination, because you only want to get the microbes from his fingers as best as possible. And he's now washing his hands, and he's doing a thorough job. Now, he did touch some things. What we do is we do this in pairs, and we have the partner turn on the water, hand the soap, that kind of thing. And we do all different kinds of soap, betadine, water, that kind of thing. So he's washing. Now, we do have sterile towels, because look, right now he's picking up towels from a dispenser. There's microbes all over that. But it's the best you can do. And he's washed his hands off. And I'm going through this as you are. I'm very excited. And it looks like now he's going to then take the lid off. Now, okay, well, he had the lid. Now he's going to do an after imprint with the fingers. And then he's going to put the lid on it, like so. And then the next step, if I can predict this, is they are now going to place that plate in an incubator, And you would do so lid side down with the auger side up to prevent water from falling on the auger plates. Oh, and it ended. Very dramatic. So we will talk about results next. Okay, we are at the end of our hand washing exercise. So we are on the very end of our outline. Some questions here. What's a surgeon trying to accomplish with a 10-minute scrub? They're trying to remove transient flora and most normal flora as much as possible. Cannot get skin sterile. It's not possible. Skin has oil. It's got folds or contours, so it prevents skin from becoming sterilized. How do normal flora and transient flora differ? Well, normal flora are permanent, part of skin and the body. Transient flora are just that. They are transient, they are temporary, they are not permanent. Now, we got a couple of things here, and I actually gave you these pictures as well, but I'm going to see if I can make these a little bigger. So what this is, these are results. Now, this looks a lot like the one that we do. Here's the labeling, the date. You always label your media as best as possible. That way you can, you don't want your results to be confusing. And you want to be able to find your plates. Here it says dirty fingertips. See where they touched? And it looks like here they touched, looks like 4, maybe 5, maybe all of them. 1, 2, 3, 4, that might be the thumb. But this is pre-washed hands. Each little circled thing there is a colony, which is like 1 million bacteria cells. Here's the dirty side. Here is after the hand washing. So whatever they did, they definitely lowered the amount of cells on skin. You can definitely see a difference there. This is cool. Now these are some big auger plates. These will fit an entire hand. They're really cool. We had some at one point, don't anymore. But you can see where they put their whole hand. Now, this is something furry. That's a fungus. And then each, see all the different colors? And like, maybe I can make this a little bigger. Get a little closer. Whoops. There we go. And see the yellow? That's a colony. That's an entire colony. Each one of those represents a different species. Like here are some flat-looking colonies. Here's a little white colony, a larger white colony. All that furry stuff there is fungus. This is a unwashed hand, and then look, if you go on the other side, a hand wash with soap and water. So they did lower the amount of microbes that were on skin. And when you do this kind of experiment, you have to be careful to always, you incubate these at the exact same time, because what will happen is if you left this plate here out at room temp for a week or in the incubator for a few days, of course this stuff is going to grow even more and would probably cover the entire plate. All right, so it looks like they worked. And yeah, that is our hand washing experiment. Thanks for joining me.